All right. I appreciate the opportunity to speak today, and thank the attendees, the organizers for inviting me and allowing the flexibility for me to present remotely. I am very excited to be presenting this case study today, and I am making certain assumptions of concepts having been already explained from the prior presenters. So if you do have a clarifying question at any point during the presentation, please speak up, and Jeremy will relay the question to me during the talk, because I don't want you to hold things to the end if it's going to prevent you from understanding what I'm talking about. So with that, I will be talking about the immunogenicity profile of Avelumab, which is the immune checkpoint inhibitor targeting PD-L1 that is being developed in collaboration with Pfizer. EMD Serono is our brand name in North America, but we are a business of Merck KGaA in Darmstadt, Germany, for those of you located in Europe. We are the same company. On the next slide is just some quick disclaimers to clarify that this drug has received accelerated approval in a couple of indications. They are not always, it's not in all countries yet, and obviously anything that has to do with treatment decisions for an individual patient must follow the labeled claims in the prescribing country. This talk is a scientific presentation of our interpretation of the data, which may or may not be in full alignment with the health authorities. Next slide. This is a quick slide demonstrating the mechanism of action for Avelumab and really all anti-PD-L1 molecules. Essentially, what happens is that when tumor cells are attacked by the immune system, they begin to overexpress PD-L1 as a means to inhibit the immune system reaction against it. So binding of Avelumab to that overexpressed PD-L1 blocks the interaction with the T cells and NK cells, with the T cells, NK cells, and then what happens is that without that inhibitory signal, the activated T cells can now more effectively target the tumor cells. And so these are often called checkpoint inhibitors because the PD-L1, PD-1 interaction is a check on the immune system. And so we're disrupting that. So it's a checkpoint inhibitor. We have three main clinical trials that were involved in the immunogenicity assessment for the Merkel cell and urothelial indication. You'll have these slides, so I won't go through it entirely. And what I'm presenting is the data that was included in the final label in the EU and in a supplement provided to the FDA, which is not yet reflected in the label. What we found when we looked at immunogenicity incidence for all subjects treated with Avelumab were that the overall incidence of ever positive was quite low. The integrated summary that we reported is 6.2% of ever positive. A minority of these are pre-existing, and we haven't seen any ADA treatment boosted in these three trials. We have subsequently seen them in a few other trials that do not have final CSRs yet. The ADA treatment emergent was 5.9%, roughly half and half between transient and persistent responses. I'll get into the definition of transient versus persistent in a future slide. Oops, sorry, I clicked onto a different page, and so I need to get back on it. Okay, the NAB is similar, except that the incidence is roughly about half that of ADA. we have 2.4% total, 2.5% for treatment emergent, which is also a reflection of a decreased denominator. And most of the NAB responses were considered persistent. When we look at pre-existing transient and persistent positive, the rules for transient are that it has to meet two criteria. One is that there is less than 16 weeks between the first and last positive sample, and that the last sample tested has to be negative. Persistent positive can meet either criteria of at greater than or equal to 16 weeks duration between first and last positive result, or the last sample is positive. Last sample positive is considered persistent because you're essentially assuming that it will remain positive in the lack of contrary data. What you can see from looking at these profiles is that most of the persistent treatment emergents are actually triggered by last positive sample rather than duration, as only 3 of the patients who had persistent antibodies actually have at least 16 weeks duration. So a lot of those assessments, which you can see on the bottom row there of those persistence are only 1 week duration, which means that they were positive at the last sample only. So then what we looked at was, well, is there a risk for subjects characterized as persistent transient to be versus transient to have differences in the titer value? And what we saw is that in general, the whole range of maximum titers was observed across the population, and that in all categories, across all subjects, the maximum titer of 1, which is the minimum titer, was the largest category, and that there was a shift in the persistent response, the first positive, that their peak was at 4, that their maximum category was at 4 rather than 1. However, in general, there was no indication from the categorization that subjects who were persistent positive had higher titers in general. Within looking at the relationship between duration and maximum titer, you can see that, again, even at the longest durations, the maximum titer was actually not greater than at the lower durations. So the distribution of maximum titer measured appears to be unrelated to the duration of treatment. So it does not suggest that the longer duration of antibodies are also related to a increase of titer over time. Measuring a potential impact on pharmacokinetics was challenging because we just didn't have a lot of patients with data both before and after seroconversion on the PK. And that is driven also by the slide I showed previously, where the challenge was that many of the positive antibody subjects are at their last sample, which means we don't have PK samples after it. So what we did to evaluate this is we really looked at the subset of subjects that do have trough concentrations following the ADA seroconversion. And what we can see is that it's kind of variable within subjects from these spider plots. It's normalized to ADA seroconversion. Some of the patients do seem to show a decrease and then an increase after seroconversion. Some of them do seem to fall and stay 0 afterwards, but there's not an overarching trend where we can say that in all subjects with ADA that they are having decreases in the PK over time. Within the subjects that have NAB, it's a smaller data set, and it's the same story again, where some of them, particularly if you look at the curve in red here, you can see that these patients, as they approach zero conversion, they pretty much go to zero, and then they don't have any time points after that are measurable. And then but for this subject with the blue stars, there's actually two subjects with blue stars, and they both follow the same pattern, which is after 80 days, after NAB zero conversion, their concentrations go up. So again, we didn't really see something that on aggregate, we would say that patients who are zero converting to NAB positive will definitely be seeing some sort of a decrease in C-trough, which is related to efficacy. The efficacy sub-analysis in Merkel cell was really done by individual patient narratives. I apologize that this is a bunch of text, but essentially the core message is out of 488 confirmed ever positives that were also evaluable for best overall response had a IERC confirmed partial response. And this 50% is actually even better than the overall trial average, but is most likely a result of the tyranny of small numbers, because 2 out of 4 is a very different scenario than a denominator of believe this study had 50 patients. Merkel cell is a orphan indication, and so the total study size is not large. What was particularly interesting is that these two patients are also quite long responders. They are both still alive, and so their overall survivor times continue to increase. But as of these data cuts, they were 19 and 27 months respectively. So safety sub-analysis is obviously a critical concern when you're looking at immunogenicity. And our first look at ADA positive samples, it uses the false assumption that ADA is a baseline characteristic, because it's actually evolving it's an outcome of the trial, just as the safety measures are outcomes. But this can be a very valuable tool in identifying the aspects of safety that are worth probing further as to whether there may be a potential causal relationship. And from looking at this graph, what you can see is that there are a few categories where there is a numerical percentage difference between the ever positive category. Oh, I just lost okay, there it is. The ever positive category in the first column versus the never positive. And those are the TEAEs leading to permanent treatment discontinuation and the IRRs, which you can see in the bottom row. In the next couple of slides, I'm going to walk you through how we then looked at particularly the IRR as sort of a case study for how you would then go through approaching this kind of a deep dive into the data. And obviously, depending on what your data is, you will come to different conclusions, or you may need to do different types of analyses than we did. The same thing was done for NAB status, and the same categories were noted of the TEAEs leading to permanent treatment discontinuation. IRRs relative to ADA seroconversion. We already knew from the integrated safety summary that IRRs were generally in the first few infusions across all subjects, and that also the ADA seroconversion was reached about 70% at approximately 4 months, and had pretty much reached 100% of what it was going to be by 6 months. So in that context, an immediate hypothesis that came to mind was, well, what is the timing of IRRs relative to ADA seroconversion? Are we looking at a subpopulation that has seroconversions, has IRRs later in the treatment regimen? Do they have higher grades of IRRs? And what we had found that in general, the ADA population was performing similar to the total population, where most of the IRRs were in the first 2 infusions. And you can see the exact numbers on the left in the text, but then we sort of grouped it into 2 rows for the table on the right, which is how many subjects had infusions, how many infusions did the subject have before they had their first IRR? 1, which means they had IRR at the first infusion, or those who had it at their first IRR at 2 or more infusions. And then looked at whether their first ADA positive was prior to or concurrent with the IRR or after. And what you can see is that a minority of the subjects had their ADA positive prior to or concurrent with their first IRR. And the vast majority of them were having their first IRR at the first infusion, and that the first ADA positive was after that first infusion, i.e. they weren't baseline positive. And so then we looked at the whether and those 3 that were prior to or concurrent with the first infusion obviously are pre-existing antibodies. The other aspect we were looking at was the maximum severity grade. So the maximum severity grade for ADA positive versus all subjects is shown in the table on the right. And what you can see is that most of the subjects still have a maximum IRR grade less than or equal to 2. The percentages are, again, numerically higher for maximum IRR grade of 3 or 4. But the tyranny of small numbers is at play in both groups here. When you're talking about 2 of 106 and 9 of 1738, those aren't large numerators, and so the percentages are not terribly accurate there. But what we weren't seeing is a dramatic shift to the higher IRR grades that were the ones that were more frequently associated with discontinuation of treatment. So to summarize, what we were looking at in the safety is that we did see a numerical increase in IRR incidence and serious TAEs, but this is limited due to the unbalanced sample size and that ADA is not a baseline characteristic. But we do not think that there is a causal relationship between ADA and the IRR occurring prior to seroconversion. We do think that there is a potential for there being a risk factor that is shared between IRR and ADA, but whatever that risk factor is not a 100% risk factor. Because there's not a perfect overlap between ADA patients and IRR incidence. And in conclusion, for the labeling, we did not see a need to monitor immunogenicity incidents separate from observing IRRs in the clinic in order to better characterize the risk to individual patients. The IRR occurrence is much more obvious than waiting for an ADA test, and the intervention would be identical whether or not the patient was ADA positive. So therefore, we didn't see a huge impact on the safety profile in general, and no change in the risk assessment. This is also influenced by the fact that the ADA incidence is quite low, that things could be different if the kinetics of ADA were earlier, and if there was a tighter relationship between ADA positivity and IRRs, either in severity or incidence. What this looks like when you then get to the labels, I have presented 2 of the labels from the ICH countries. The top one is for the approved USPI. This actually contains a smaller data set of the subjects. And the lower box is the immunogenicity language from the approved SMPC in the EU. And this is the data that matches the presentation. What I'm showing on this slide is actually not necessarily the actual incidence numbers, because those are different. However, I did want to bring up that just because this is my scientific interpretation as an immunogenicity expert within Merck /EMD Serono, we do have different interpretations by the two regulatory authorities. So if you focus on the last sentence here on the immunogenicity of in the USPI, oops, I just realized I clicked to highlight it and that advanced the slide. Here they agreed with our assessment that the development of treatment emergent ADA did not appear to alter the PK profile or the risk of infusion-related reaction based on the data shown. However, in EU, they have not fully agreed with our assessment on ADA, where they are, and IRR, where they say there may be an increased risk for infusion-related reactions. So, of course, this is a topic of continued discussion with EMA, as what we will be continuing to provide updated integrated immunogenicity assessments, including additional monotherapy trials, where we are continuing to generate data on this potential risk. And so far the data has remained consistent with what I have shown you today. The other big difference in these labels is that we did not have the neutralizing data at the time of the US label, and we do have it at the time of the MAA. But given the very limited data on neutralizing antibodies, they were unwilling to include anything on the incidence of neutralizing antibodies in the label at this time. Since it doesn't affect the treatment regimen, that's acceptable, and we will be looking to continue to update this portion of the label, concurrent with other label updates to include additional indications or other key findings that may evolve as the drug is used in the commercial setting. So the summary across all of the analyses for Avelumab is that we as in the sponsor, feel that the development of treatment emergent ADA against Avelumab did not appear to alter the PK profile, risk of infusion reactions, or maintenance of the response in individuals who seroconverted. The low incidence is repeated here, that we didn't feel there was a clinically meaningful impact on PK. The partial responses were maintained after ADA seroconversion, and that the overall risk-benefit assessment was unaffected by the presence of ADA in the overall population, and that the timing of including that the timing of IRR is early and primarily prior to seroconversion. I'd like to acknowledge an extended group. As anybody in industry knows, it takes a village or sometimes a major city to complete analysis of immunogenicity in the context of the overall clinical data. Within my immediate department, I would like to acknowledge several critical people, including the operational staff, my peers within the immunogenicity expert group, the experts who prepared the PK by PD and PK by ADA analyses, and our senior management. This is, again, it's a collaboration between EMD, Serono, and Pfizer. Core groups that we have worked with include drug safety, global biostatistics, and data management. And I would also like to acknowledge our external bioanalysis partners, which include Covance, QPS, and Quintiles, for different aspects of the data included in this presentation. With that, I would like to thank you for your attention, and I am happy to take questions at this time. I do realize I ran over due to us also being started a little bit late, So I would defer to Jeremy in terms of how many questions we can take at this time.
Thank you, Joleen. Thank you very much.